Okay, good afternoon, everyone. So we'll go ahead and get started. So first I want to welcome each and every one of you to the 18th annual, just admitting someone else, 18th annual Summer Student Research Symposium. This year we're doing it in virtual format for the first time. So the symposium showcases the work of students in the Emerging Caribbean Scientists Program, also known as the ECS program, which offers scholarships, summer programs, research experiences, mentoring, and supplemental instruction to UVI students who are majoring in science, technology, engineering, and mathematics, also known as STEM. So programs within the ECS program include the Maximizing Access to Research Careers, also known as MARC. We also have the Historically Black Colleges and Universities undergraduate program, and we have the Research Initiative for Scientific Enhancement, also known as RISE. There are also summer undergraduate research experiences, and then the Summer Sophomore Research Institute. The University of the Virgin Islands is also one of the 10 minority-serving universities for cooperative agreement awards with NASA under the Minority under University Research and Education Project. There is the Cybersecurity Fellowship Program at UVI, whose goal is to provide training and in the future increase the number and diversity of the nation's cybersecurity experts. In addition to these programs, this year's summer research is supported by the National Science Foundation, the College of Science and Math, and the generous donations of private donors. So at this symposium, we have 6 sessions that are being ran concurrently. You can find the schedule in the abstract book and also on the UVI ECS website. We will keep presentations on schedule to allow symposium attendees the opportunity to move between different rooms. We would like to stick to schedule, so if there's a student who's missing, the next presentation would not start until the scheduled time. So if you are unfamiliar with Zoom, take a moment to familiarize yourself with its features. We ask that you remain on mute throughout the presentations and put any questions you might have in the chat box. And just to remind you, in case you're not sure where you're at, this is session 3 for the Biomedicine and Community Health section. So my name is Dr. Lavita Rogers, and I'm with Dr. Verlene McSween, and we're the moderators for this section. Okay. So first up, we have Naomi Douglas, and we have a pre-recorded video from her, but she's also present to answer questions once her video is finished. So I'll start playing that video exactly at 1:10. So just another quick note to the presenters. So you have 5 to 7 minutes to present, and then after that, there's a 2 to 3 minute session for questions. Just to remind you, and then look out in the chat box. I will send you, for those of you who are not doing a pre-recorded presentation, look in the chat box for a notice for when you're at the 5-minute mark. Okay, so it's 1:10. So we'll get started with Naomi Douglas and her presentation.
Good afternoon. My name is Naomi Douglas. I am a biology major and Mark scholar. And with my mentor, Dr. Rogers, I studied the meta-analysis of gene expression data. What is tuberculosis? Tuberculosis, or TB, is a lung disease caused by Mycobacterium tuberculosis, which is shown over here. This disease is one of the top 10 causes of death, and the symptoms may include cough with occasional blood, chest pain, weight loss, and fever. The TB bacteria is spread via airflow from person to person, and it is dispersed into the air when the infected person or persons cough, sneeze, or simply speaks, which is shown in these figures here on this slide. When a person breathes in the TB bacteria, it can settle in the lungs and can move through the blood to other parts of the body, such as the kidney, spine, and brain. According to the World Health Organization, people who are infected with HIV are 19 times more likely to develop active TB because of their weakened immune systems. Alcohol use disorder and tobacco smoking increase the risk of TB disease by a factor of 3.3 and 1.6 respectively. And over 95% of cases and deaths are in developing countries. As you can see here from this figure, there were 1 million children that fell ill with TB in 2017, and there were 233,000 child TB deaths in 2017 as well. My research question was, how does disease state, age, gender, and ethnicity affect gene expression for tuberculosis infections? To answer that question, my objectives were to understand how disease state, age, gender, and ethnicity affect gene expression responses to tuberculosis through expression data, and to reuse public data to answer new questions. The first method was data curation. I collected data sets on tuberculosis patients, focusing on their disease states, ages, genders, and ethnicities. Data was organized and integrated using Gene Expression Omnibus, or GEO, which is a public functional genomics data repository that stores array and high throughput data. I originally created 6 data sets and a total of 294 samples in total, which is shown here in the summary of data sets. These are the accession numbers, the number of males and females, and their age ranges. The second method is data preprocessing. This is where I downloaded the raw expression data and preprocessed it by performing background correction, normalization, summarization, and probe annotation. So onto our third method is data analysis. To identify genetic variation in the data across ages, genders, race, and disease states, an ANOVA was performed. This is a summary of the ANOVA. I compared all pairwise interactions between factors being the study, age, sex, disease status, and race. And here is a heat map. So this heat map shows expression of the genes across the samples, and I will conduct downstream analyses to assess regulation and whether genes are up or down regulated based on disease status. So here shows the top 10 differential genes expressed. I researched the genes and saw their functions. So their functions were that ANGSA3 possesses anticoagulant properties. ABHD10 is a metabolite of the immunosuppressant drug mycophenolate. CLEC4D is involved in antigen uptake at the site of infection, either for clearance of the antigen or for processing and further presentation to T cells. CLP1 and LPCAT3 has catalytic properties for mRNA formation. MAX is a transcription regulator. UNC119 plays a role in trafficking proteins in photoreceptor cells. ANKRD35 functions in RNA regulation. DUSP8 functions in gene silencing. And ZNF331 functions in transcriptional regulation. So these genes have functions in gene regulation and gene expression. So we found 1,790 differentially expressed genes by disease factor, with the p-value being less than 0.05. Of those 1,790 genes, 2 genes, ASO1 and AGO2, had disease and age interactions. So ASO1 functions in protein binding and Angel2 in RNA regulation. There were 34 genes that had disease and race interactions, and we found no genes that had disease and sex interactions. The final method is pathway and gene enrichment analysis, which helps to find out whether the differentially expressed genes were associated with a certain biological process or molecular function. So this is a table of the enriched gene ontology terms, so the biological processes. We have here reproduction, metabolic process, cell killing, and immune system process, and these are its gene ratio. This is the top reactome pathways by significance. The infectious disease reactome pathway was also enriched and was number 11 on the list out of a possible 90 pathways ordered by statistical significance. These are my broader impacts. Minorities, mainly Blacks, according to data obtained, that have historically experienced greater obstacles to health are specifically affected by TB. Our findings highlighted potential gene targets for TB and genes to further explore how race and age may influence disease outcomes. In the future, we can provide biomarkers or new targets for vaccines or treatment of TB given age, race, or sex of the person once the expression profile is examined. So there were challenges faced, mainly from the data creation. So data was not reported uniformly, the data was missing, some details were unclear, and the sample size. So out of the original 133 data sets that were collected from the GEO, 5 data sets were used to gain results. So some data sets did not mention every factor like the age, sex, disease state, and ethnicity. And we also focused on the Illumina microarray platform. So that caused the number of male and female samples to be unequal. Another challenge faced was that the limitations to using microarrays expression data. So we only studied the annotated genes because we could not study and explore the novel genes like with the sequencing. In the future directions, we want to find out what genes are up or down regulated. We want to perform another analysis to determine how genes are regulated given the different factors, and we want to curate more data. These are my acknowledgments. The funding was provided by NIH, MARC at UVI, a holistic approach. And these are my mentor and I's contact information. So if you have any questions.
Okay, we'll open the floor now for questions. Okay, so there's one question for you, Naomi. So do you expect to see similar changes in the same genes in other respiratory infections? Or are these patterns of gene expression specific to tuberculosis infection?
These patterns of infections are specific to tuberculosis. So when I did a data curation from the GEO, I specifically searched for the tuberculosis patients.
Okay, thank you. Any more questions for me? No? Okay, well, thank you, Naomi. So next up, we have Elena.
Good afternoon, everyone. My name is Elena Willis. I am a rising sophomore and a biology major here at UVI. This summer, I've been working alongside Dr. Lavita Rogers on a meta-analysis of gene expression variability in asthma. Next slide. So asthma is an inflammatory disease of the airway. If you look at the top right image, you'll see that asthmatics tend to have narrower and inflamed airways. Now this leaves them susceptible to what's commonly known as an asthma attack. And asthma attacks usually present as with coughing, wheezing, chest tightness, and a shortness of breath. Now, according to the CDC, there are over 339 million people worldwide who currently suffer from asthma. It is also the most common chronic disease affecting children. Asthma is also a multifactorial and a heterogeneous disease, and so that makes it quite a complex and interesting disease to study. Next slide. So for my research, I used publicly available microarray expression data, and I reanalyzed it to identify differentially expressed genes in asthmatic individuals compared to healthy individuals. And so my specific research question this summer is, how does sex and disease state affect gene expression for subjects of asthma? Coming into this project, I hypothesized that that gene expression would be influenced by sex, and that's why I selected sex as a factor. So this is an overview of my methods. I first conducted a data curation, and I selected for the 4 data sets that I wanted to analyze. I then pre-processed that data in R and conducted an analysis of variants, which identified the differentially expressed genes. I visualized those genes in heat maps, and then I conducted a few gene and pathway analyses. Next slide. So our analysis of variants identified 1,641 differentially expressed genes, and that's just looking at disease state as a factor. We also analyzed further for genes that were influenced by sex, and we found over 9,000 genes. However, because the majority of these genes are associated with sex-related differences like hormonal differences, we concluded that there is no interaction between disease status and sex. So on the right is a heat map that we generated of the top 10 differentially expressed genes. On the left, you see some associations of 3 of these genes. So the CLC gene has been associated with allergic rhinitis, which is when the eyes start to tear up and the nose becomes inflamed in response to an allergen. CLC is also associated with hypereosinophilic syndrome, which is where you have an elevation of eosinophil in the blood. Eosinophil is a type of white blood cell that's very important in our immune system. It causes the inflammation and the dysfunction of the cells that it targets. Now, RNA-SE2 gene has been associated with pulmonary eosinophilia, and that's when you have a infiltration of eosinophil in the lungs. And what this does is it causes the airways to constrict, and that's also a phenotype that we found is associated with asthma. So the CDC gene, the CDC1-3 gene, is associated with Wolfram syndrome 1 and Wolfram syndrome. And these are both neurodegenerative diseases. So some symptoms include loss of vision and high blood sugar. Now, as of now, I'm not able to state how it might be associated with asthma, but I do know that like the rest of these genes, they did have a significant difference in gene expression based on disease state. Next slide. So we then conducted a Reactome analysis, and the results, it showed that our differentially expressed genes were enriched in these 4 pathways as seen on the right. Neutrophilic granulation, it had the highest gene count. It was also the most statistically significant pathway that we found. So neutrophils are a type of white blood cells. If you look at the image to the bottom left, you see that they contain these granules. So in response to, for instance, a bacterial infection, these neutrophil cells will degranulate or release the contents of these granules, which are usually antimicrobial and pro-inflammatory proteins. So once again, we're seeing that connection with asthma with regards to an inflammatory response. Next slide, please. So we also conducted a Kegg pathway analysis, and we identified these 4 gene ontology terms. So hematopoietic cell lineage, that's when you have stem cells that give rise to a variety of other blood cell types, such as neutrophils and eosinophils, which, as I previously discussed, play a huge role in our inflammatory and immune response. The Kegg analysis also identified glioma, which is a type of brain tumor that's one study found actually had an inverse association with glioma and a history of asthma. A similar finding was also found with regard to prostate cancer, where they found that that men who had a history of asthma were actually 29% or about 29% less likely to develop prostate cancer. Next slide, please. So yeah, with these genes and pathway associations, our goal, our plan is to improve our understanding of the molecular pathology of asthma. Hopefully we'll then be able to create genetic testing that could even be tailored towards identifying children that are at risk, higher risk of developing cancer. Also, we could use our findings for the purposes of gene therapy, which would then prevent the onset of cancer by influencing or by changing our gene expression. So my next steps are to conduct a post-hoc analysis, which will identify which of these differentially expressed genes are being upregulated and which are being downregulated. I also want to continue my data creation, excuse me, to conduct an age-specific analysis and to also improve my sample size and the statistical significance of my results. Next slide. So I would like to acknowledge the UVI RISE program by means of the NIH for funding this research. And I'd also like to give a special thank you to Dr. Levita Rogers for her guidance and mentorship this summer. Are there any questions?
Okay, so I have 2 questions for you. Oops, I'm showing up in 2 places, sorry. Okay, so why did you choose gender as your variable as opposed to race, for example?
Yeah, so I did initially want to do a lot of factors, including, as you said, race or ethnicity, as well as, sorry, age and obesity and a variety of things. But I was limited because I was using publicly available data. And so not a lot of the researchers specified or reported the individuals or the patient's ethnicity, for example. And so I did not have those factors to consider.
Okay. What physiological relationship could there be between the development of asthma and the protection against the development of a glioma?
Oh, okay. So this isn't confirmed, but I did suspect that, you know, as I mentioned, asthma has a lot to do with an increase in these white blood cells like the neutrophils and the eosinophils. And, you know, their role in our body is to target invaders or abnormal cells such as cancer cells. So it is possible that that could reduce a person's chances of getting these two cancers.
Okay, and then the last question. So we have some psychology majors and people that might not know what a heat map is. So can you describe what a heat map is? What do you mean by Kegg and Reactome pathway analysis, and what are they used for?
Oh, okay. So the heat map, basically what you should get from the heat map is that it's just showing the intensity of expression of these genes. And so the darker the shade, the more overly expressed that gene was. On the side, all the pyrographic kind of connections were just showing connections between the genes. And the analysis that I did, they're just looking at associations. So they're looking at all of these genes, which like, let me connect this. So you have different pathways, and the genes that I put into this analysis, it's, I don't know. Okay, so it showed that it resulted in the pathways based on like which genes were more present in that pathway. I don't know if that makes sense. Like out of all the genes, which one was most abundant in that pathway, I guess. I don't know if that makes any sense. What was the question again?
So the question is basically just saying, so you did a Kegg pathway analysis.
Yeah.
Why do you do that?
Oh, just to identify what pathways these genes were associated with.
Yeah. Okay, and Dr. Stanford has a question.
Mm-hmm.
Hi, good afternoon.
Good afternoon.
So I was wondering, because I know a child who has really severe asthma, dangerous asthma.
Mm.
Can research like this help him?
Yes, to a certain extent. I didn't go much into it, but asthma is also, as I said, it's multifactorial and it's very complex, as in there are many genes that are associated with it and many factors, environmental factors like pollen and allergens and so forth. And so what all my research aimed to do is to identify or help us understand a little bit more how these genes are affecting the onset or development of asthma. And hopefully, yeah, as I said, we might be able to predict it with the genetic testing, but then again, you also have to take into consideration that there are a lot of environmental factors that also play a role in the onset of asthma as well.
So if you do the genetic testing and,
Yes.
and you find the genes that are responsible for his asthma, does that help design treatments?
Possibly. Possibly. I mean, gene therapy and other sort of treatments like that are, they're not, they're far in the future, I guess, until they're perfected. But it is possible that once we identify these genes, we might be able to influence the gene expression, in the person's DNA. Yeah.
Have you seen any of these articles about gene therapy for sickle cell anemia?
No, I have not.
You should look that up. It's really cool.
Thank
They
you.
have cured people. Just a couple of people, but.
Okay.
Thank you.
Yeah, thank you for the questions.
So next on the schedule is Deidre Lee, but the student is missing. So we'll just have, I guess we'll have a short intermission until 1:40 for the next presenter. Okay, everyone, so we'll continue. Next up, we have Kaisha Jean-Baptiste, and she also submitted a pre-recorded presentation. So I'll start playing that right now.
Good day. My name is Kaisha Jean-Baptiste, and today I'll be presenting the research paper, Late Viral or Bacterial Respiratory Infections in Lung Transplanted Patients, Impact on Respiratory Function. So in recent years, lung transplant survival rates has increased due to medical improvements and better selections of recipients and donors. Regardless of this, there are some things that also affect the overall health of these patients. So primary graft dysfunction or acute rejection has been found to be the cause of early deaths in these patients. Infectious diseases are also responsible for about 1/3 of post-transplant deaths within a year. And it has also been found that bacterial infections are the leading cause of respiratory infections in lung transplant recipients. Acute rejection and graft dysfunction are not the only things that affect the health of these patients. There's also bronchitis obliteration syndrome, which affects about 45 to 75% of transplant recipients. So the goals of this presentation were to assess the occurrence of late viral and bacterial respiratory infections in LTRs, lung transplant recipients, and to compare their impact on graft function with those without any respiratory infections. The hypothesis here was that the impact of viral infections is at least as severe as bacterial respiratory infections on lung graft functions among LTRs. To do this paper, patients were chosen that had received lung transplants, and respiratory samples were collected from them. Once these samples were collected, the patients were divided into 4 groups. This flowchart shows how the patients were separated into their groups, with about 53 being excluded because of death within 6 months or no respiratory evaluation 6 months after inclusion. So the groups that these remaining 99 patients were separated into included the viral infections, the bacterial infections, which included both viral and bacterial infections, and patients with just bacterial infections, and the control group. For all statistical tests completed in this paper, the VIG, BIG, and CG were all compared, and there was found to be a significant difference in the acute rejection treatment for each group. There was also found to be a significant difference in VOS at inclusion for the VIG group compared to other groups, as well as a significant difference for only one symptom between each group. So the overall results of this presentation were that for most symptoms and prior viral infections, they were similar across all 3 groups, but more prevalent in the VIG group, and that acute rejection requiring treatment in the 3 months prior to inclusion was needed more for the VIG group patients. There was also a higher occurrence of BOS at inclusion for the viral infectious group. This second table shows the symptoms that were observed in patients in both the viral infectious and bacterial infections group. And it shows that for the bacterial infectious groups, the patients suffered these symptoms more and required more hospitalizations than the viral infectious groups. This third table shows that there were some viruses and bacteria that affected the viral infectious group and the bacterial infectious group, with the P. coronavirus affecting both groups more, and the Pseudomonas aeruginosa affecting both groups. The major conclusions for this presentation were that the major conclusions in this presentation were that in accordance to the hypothesis, viral respiratory tract infections can be as severe as bacterial respiratory infections. Viral respiratory infections and virus bacteria co-infections were found to be more frequent in lung graft recipients. Viral infections were strongly diminishing initial lung function and often led to more hospitalizations. And single viral respiratory infections led to greater deterioration in lung function and more acute rejection than bacterial infections. There were some limitations in this paper where there were a small number of patients, especially for some categories and tests. And because of that, there was a need to merge the bacterial infections group with the patients that had both bacterial and viral infections. There were also some biases within the experimental design because there were a higher infection detection in patients with AR or BOS due to more frequent visits 3 months prior to inclusion.
All right, so now we transition to the questions. Okay, so we have one question for you so far, Kaisha. So do you think the body responds differently during bacterial versus viral infections? If so, why?
I do think the body responds differently to bacterial versus the viral infections. And I also think that that might be the case because some of those viral infections were things that the patients had already experienced before. So they might have already been treated for those viruses. So their body might have already had that training basically for that virus. And it might not have been the same thing for the bacterial infection. So that's why I think that's different.
Okay, and then why do you think patients were most infected by the specific viral and bacterial type mentioned?
I'm not exactly sure why that specific bacteria was the ones that patients had the most infections with. I want to believe that that might be something that is because of their environment. They might be exposed to different things. I think this paper was done in Paris, so that might be something that just came about from exposure in that particular place.
Okay. Did they explore like the state of their immune system? Like, did they talk at all about whether they were like, I know some of them were like they had like lung transplants or lung grafts, but did they talk about whether they were all generally healthy or anything like that?
They didn't say exactly if the patients were generally healthy, if they had any type of problems with their immune system. They didn't really say anything about that in this paper.
Okay. All right. Thank you. Let's thank Kaisha. So next up, we also have a speaker who's missing. His name is Sequoia George. So we'll have another short intermission until 2 o'clock. Okay. Okay, so we'll continue with our next presenter. It is Alkana Akuna. So I hope I said your name correctly. So she also sent a pre-recorded presentation. So let's go ahead and listen to her.
Good day, everybody. My name is Alkana Akuna, and today I will be talking about could easy access to fast food restaurant lead to childhood obesity. To give you a brief background, what is obesity? Obesity, according to Mayo Clinic, it is a complex disease involving excessive amount of body fats. The method to determine whether a person is fat is they use the BMI, or known as the body mass index. Obesity can occur at any age. Yes. So they define fast food as food service outlets quickly serving inexpensive foods in minimal preparation and table service. The article is called Weight Gain in Mid-Childhood and its Relationship with the Fast Food Environment. This study was done by Matthew Pierce in 2017. Their purpose was to explore associations between weight gain in children and fast food environments. The methods. The sample size, they have total of 1,577, 827 are boys, and 750 are girls. The data are collected from NCPM for South Gloucestershire, England. This was taken during 2006, 2007, and again on 2012 and 2013. The data for fast food was taken from the health department. The statistical analysis they use are geographic information system, the Shapiro-Wilk test, mean and standard deviation, the t-test, and the Spearman-Rank correlation coefficient. And here on figure 1, they found out the total of fast food identified in South Gloucestershire is 175. As you can see here, in the red, that's where the fast food outlets are located. Table 1 shows the mean accessibility score for all children was 0.74. And the mean score for boys are a bit higher, which is 0.76 compared to girls, which is 0.71. But there was not a strong difference. In figure 2, there was a strong correlation of p-value of 0.01 between the number of fast food outlets per 1,000 population by deprivation. 32.7% of children were found not to have any fast food outlets within the 1 kilometer of their postcodes. In figure 3, they use a Spearman correlation to access the relationship between the children's deprivation score and the accessibility score. They found the p-value of 0.01. The data suggests that children with higher accessibility to fast food outlets have increase of 89% compared to the children that have least access to the fast food outlets. The areas with the highest prevalence, which is the red here, they are more likely to have a dense fast food outlets. They found that there's a greater density of fast food outlets in a lower income neighborhood than the wealthier neighborhood. So probably the wealthier are around here while the lower income lives around this area. The reason for that is an equal distribution of fast food outlets. So to conclude this presentation, this study support the theory and new evidence that environment may play an important role in the development of obesity. Children who have greater access to fast food outlets were more likely to gain significant amount of weight. Some of the limitation of this study, first is the use of postcode to determine the child's residence in the fast food outlets. They probably might have a slight miscalculation when doing this. The second one is that they did not include the other places or the neighboring of the neighboring area within the Gloucestershire. Also, the mapping of the fast food, they only did the mapping during 2014. So therefore, they didn't know if there's any changes from 2006 to 2013. So for the future studies, they could develop a study where location, ethnicity, and socioeconomic status are all included. And they could also compare and contrast fast food versus dining restaurant to see which one is more prevalent to weight gain in probably adult. And these are my reference, and that concludes my presentation. Thank you very much.
Okay, so I was just checking and I do not actually see that Elkanah is present. So if you do have questions, you can go ahead and send them to me and I will email them to her and so she could answer us at some point. But so we will take like 4 minutes, 5 minutes before we get to the next speaker so that we're sticking to the schedule. Next up, we have Alexan Carr. But if you do have any questions, please feel free to type them in the chat for Elkanah. Okay, it's 2:10, so we'll move to Alexander. So we have another pre-recorded presentation. So as you think of your questions, you can type them in the chat.
Good afternoon. My name is Alexander Carr, and I am a Rice Scholar and sophomore psychology major at the University of the Virgin Islands. I worked under the guidance of Dr. Aletha Bauman this summer to see if blood pressure and perceived racial discrimination may be a predictor of episodic memory among older Black Caribbean adults. Do blood pressure and perceived racial discrimination predict late life episodic memory in older Black Caribbean adults? Blood pressure and perceived racial discrimination do predict late life episodic memory among older Black Caribbean adults. Have you ever wondered, where did I just put my keys? Where did I park my car? Why am I even in this store right now? All of this is episodic memory, which was first discussed by Endel Tolving in his 1972 book, Organization of Memory. Tolving described episodic memory as a recently evolved, late developing, and early deteriorating past-oriented memory system, more vulnerable than other memory systems to neuronal dysfunction and probably unique to humans. This memory system gives humans the ability to remember past experiences, our own personal one-way time machine embedded in our brains. Blood pressure is a composite of two pressures. We have systolic pressure, which is the pressure against the artery walls as your heart beats. And then we have diastolic pressure, which is the pressure against artery walls as the heart is resting between beats. Each participant in this study had 2 blood pressure measures taken. I will be getting the mean arterial pressure by using the equation systolic blood pressure plus 2 multiplied by diastolic blood pressure divided by 3. These results will be recorded as millimeter of mercury, mmHg. The American Psychological Association Dictionary defines racial discrimination as the differential treatment of individuals because of their membership in a particular racial group. It has also been shown that extended exposure to experiences of discrimination have negative consequences on cognitive decline later in life. My participants are all between the ages of 50 and 80. They are non-smokers. They are fluent in English. They identify as African American, Black, or Afro-Caribbean. They have no current diagnosis of a neurological condition, nor are they on any medications to treat a neurological condition. They have resided on St. Croix for the better part of 30 years, and their vision needs to be able to be corrected with glasses or contacts. We dispersed the information about this study by disseminating flyers and postcards to businesses throughout the St. Croix community. Word of mouth was used at the February 2020 Agricultural Fair. And due to the fact that we do not have a computerized list that we can randomly select participants, convenience sampling was used. A sphygmomanometer was used to measure blood pressure. The National Alzheimer's Coordinating Center, UDS, Neuropsychological Battery 3.0 was used to measure multitudes of cognitive areas. Out of that, episodic memory was measured using the Craft Story 21 recall, immediate and delayed, which is the research assistant reads a story to the participant, and the participant repeats back what they remember immediately. And then after a 20-minute delay, they are asked again if they remember that story. Also in the NAC is a category fluency. And this is where the research assistant asks the participant to name as many animals or vegetables in a 1-minute time period. Other measures of episodic memory are the mnemonic similarity task, the MST, which is a two-part computerized assessment, and also the Ray Auditory Verbal Learning Test, which is two-word lists read a number of times by the research assistant, and the participant repeats the lists. And then after a 20-minute delay, the research assistant then reads another list of words and asks the participant to identify whether those words were on the first two lists or are they new words being added. Perceived racial discrimination was measured using the index of race-related stress. This questionnaire covers cultural, institutional, individual, and collective racism. On March 12, 2020, all in-person data collection was suspended by the Institutional Review Board at the University of the Virgin Islands due to the COVID-19 pandemic. This suspension is still in effect, causing further delay in collecting and analyzing data. During the fall 2019 semester, both undergrad research assistants, Azrael Williams and myself, were trained to collect data, and we also made recruitment plans to use posters and flyers created by the research team. Due to the size of this study, 2 visits were required to be able to collect all of the data. We currently have 6 participants that completed visit 1, and we have 3 participants that completed both visit 1 and 2. 1 participant was not eligible to proceed to visit 2, and we have 1 participant that was not scheduled before the suspension of in-person data collection. Here are my references. Do we have any questions or comments? I would like to thank you all for listening to my presentation today. And I would like to take time to thank the Emerging Caribbean Scientists Program, the Alzheimer's Association, Boston University, UVI, Dr. Karen Schoen, Dr. Aletha Bauman, Michael Rosario, and Azrael Williams.
Okay. So now we'll go to the question section. Okay, so there's one question. So how do you separate out the effect of poverty or other factors related to race from perceived discrimination?
How do I separate the poverty?
Yeah, so how do you separate out the effects of poverty or other factors related to race from perceived discrimination?
Well, part of the bigger study under Dr. Karen Schoen and Dr. Aletha Bauman and Michael Rosario, they do a socioeconomic status questionnaire, but I didn't include that in my study. So at this point, I couldn't tell you how I would go about that, but you could follow up with me and I would be able to find that answer for you.
Okay, sure. Yes, we could do that. Okay, another question. If this episodic memory or dementia is related to perceived discrimination, do you think psychological interventions would reduce the development of episodic memory? I can say it again if you need me to.
Yes, please repeat. I'm sorry,
No problem. So if this episodic memory or dementia is related to perceived discrimination, do you think psychological interventions would reduce the development of episodic memory?
It could be that a therapeutic approach could help with dealing with perceived discrimination, racial discrimination, because it's our perception of it. So it's more of a personal question of, do you feel like you were treated differently because of your race just now?
instead of me asking the person who was doing the treatment, are you treating that person differently because of their race? So I couldn't imagine that therapeutic steps wouldn't be helpful, but I am not 100% sure on that.
Okay. Thank you. So
Thank
we'll
you.
move, no problem, thank you. So we'll move to the next speaker. So now we have Ashley Challenger, and she'll be talking about intimate partner violence. So I'll go ahead and press play.
My name is Ashley Challenger, and my mentor is Dr. Althea Bauman, and today I'll be presenting intimate partner violence. The title of my study is Depression and Prevalence of Types of Intimate Partner Violence Against Afro-Caribbean Women in St. Croix and St. Thomas. The content that I intend to present to you today is my research question and hypothesis, the null and alternative statistical hypothesis, the participants of the study and the instruments used to gauge the scales of abuses, the method and the data analysis portion. Moving straight ahead into my research question, what are the relationships among types of intimate partner abuse, depression, and residents of Afro-Caribbean women in the Virgin Islands? For my research hypothesis, I have three for the three variables that I looked at throughout this study. The first is physical and sexual abuse has a higher likelihood of leading to depression than emotional abuse. The second is physical and emotional abuse has a higher likelihood of occurring in St. Croix rather than St. Thomas. And the third is that depression rates are likely to be higher on St. Croix than St. Thomas. Moving into my null and alternative hypothesis, the null is that there is no relationship between depression and type of abuse, there is no relationship between type of IPV and island, and that there is no relationship between depression and island. The alternative hypothesis is that there is a relationship between depression and type of abuse. There is a relationship between type of IPV and island, and that there is a relationship between depression and island. Some of you may be wondering, what is intimate partner violence? Intimate partner violence is defined as the physical and sexual violence, stalking, or psychological harm by a current or former partner or spouse. Moving into the participants of this study, the women from this study were recruited throughout the years of 2009 through 2012, and they were recruited primarily because of a visit to any primary prenatal or family planning clinics based on either island. The women had to meet certain requirements to be included in the study. They had to be within the age range of 18 to 55. They had to have reported being within an intimate partner relationship within the past 2 years of the study, and they had to identify themselves as being of African descent. Combined, the women that were recruited were 545 from the island of St. Croix and 514 from the island of St. Thomas. Moving into the instruments that were used to scale the types of abuses. For sexual abuse and physical abuse, the severity of violence against women scales were used. For sexual abuse, if the women responded positively to sexual questions that pertain to forced sexual activity or intercourse, they were deemed sexually abused. If they responded positively to questions that indicated that their abuser is a current or former partner, they were deemed to be physically abused. To scale the psychological abuse, the women's experiences with battering was used, and if a woman scored a 19, she was considered to be psychologically abused. For depression was scaled using the 20-item Center of Epidemiological Studies depression scale. Moving right along to my method, the data that was used was from the NIH-funded Abuse Status and Health Consequences for African American and Afro-Caribbean Women. This was a grant that was given to the university's Caribbean Exploratory Research Center. I did receive permission from Gloria Calwood, who was the principal investigator at the time, so as I could access the de-identified data on the three variables that I used throughout my study. Permission to the current director, Noreen Michael, is still pending at the moment. The research was approved by IRB, and it was conducted by researchers from the university's Caribbean Exploratory Research Center and stateside universities. Moving right along into my data analysis, for descriptive statistics, my aim was to get the percentages of women from each island by the type of abuse they sustained, and to then see if that was a predictive factor in the likelihood of depression. Combined, 31% reported experiencing only emotional abuse, 46% reported experiencing only physical abuse, and 54% reported experiencing physical and sexual abuse. For my inferential statistics, the chi-square of independence was used, and after that was conducted, I concluded that there was no significance between the type of abuse and island, there was no significance between IPV by island, and there was no significance for depression by island. However, I found that there was significance in type of abuse and depression by IPV, though both of those relationships were very weak. Moving straight into descriptive statistics specifically, the women who reported both physical and sexual abuse, which was a 54%, are more likely to experience depression compared to those who reported only physical abuse and only emotional abuse. Straight into the influential statistics, for intimate partner violence by island, which was not significant, the women in the territory, both St. Thomas and St. Croix, combined 32% reported experiencing IPV within their lifetime. Intimate partner abuse by island, which was also not significant. For St. Croix, 70% reported abuse, while at St. Thomas, 65% did. While I was conducting the study, I did find that there was a trend between intimate partner abuse at island, but that there was nothing significant about it. For depression by island, which was also not significant, 2/3, which was 68%, reported being depressed. And that came from the 1,090 women who were included in that study. For depression by intimate partner violence, which was significant but showed a very weak relationship, 54% of women who were abused also were depressed, while 42% who were not abused at all were depressed. These are the references that I used throughout my presentation today. And I would like to say to anyone, to those who are listening, that if you or anyone that you know find yourself within an intimate partner violence relationship, you are not alone. And there are different organizations placed on both islands to help you with any questions or concerns that you may have, or to help you get out of the situation that you are in. I would like to end by thanking everyone for being here today and for taking time out of your day to listen to my presentation. And my last slide includes the different organizations placed on both islands that you can reach out to if you find yourself in an intimate partner violence relationship, or if you know someone who is in one. For St. Croix, it is the Women's Coalition, and for St. Thomas, it is the Family Resource Center. If you find yourself in an emergency, 911 is always the option.
Okay, so moving right into the question section. So I have two questions that are both asking the same thing. So the question is, why did you hypothesize that the intimate partner violence would be greater on St. Croix compared to that on St. Thomas? And did you have data or anything to suggest this?
Initially, I did not have data to suggest it. It was just the way that I felt heading into the study. So that was the best choice for me.
So was it that you thought that males or
I thought that it would be more dominant on St. Croix than on St. Thomas.
Okay. Did you have any specific reasoning for that you would like to share?
I would base that based off of word of mouth of certain situations that have happened on the island of St. Croix. I will say I've lived on the island of St. Croix for quite some time, so it was just my own opinion that I brought into my hypothesis that I wanted to figure out for myself. I wanted to answer.
Sure. Okay. And then just one last question, just to clarify what was considered emotional abuse.
Pardon?
Just one last question to clarify what was considered emotional abuse.
Emotional abuse was along the lines of, you could say verbal or psychological.
Okay. All right. Thank you.
You're welcome. Thank you.
Okay, good morning and good afternoon, everyone. My name is Brenna Anderson, and today my partner Brenna Crummell and myself will be presenting on racial disparities in prostate cancer research. Our mentor is Dr. Bahari.
Prostate cancer is cancer of the prostate gland, which is responsible for semen production as well as the nourishment and transportation of sperm. The prostate is located below the bladder and in front of the rectum, as seen in this image on the right, which displays a normal prostate gland on the left and a gland with active cancerous cells on the right. There are usually no symptoms. However, someone with prostate cancer may present symptoms such as trouble urinating, blood in semen, or erectile dysfunction. There are no clear causes for the disease, but it is evident that prostate cancer occurs due to cells in the prostate becoming abnormal. In addition, factors such as age, race, and obesity can increase your risk of developing prostate cancer.
Could you press the space bar again 3 extra times? Okay, thank you. Okay, prostate cancer and race. 1 in every 4 Black men is likely to be diagnosed with prostate cancer throughout his lifetime, 1 in every 8 white men, and 1 in every 20 Asian men. Black men are twice more likely to die from prostate cancer if diagnosed, and 76% more likely to be diagnosed when compared to white men. All of this raises the question of what is the reason for this great level of racial disparity between Black men and men of other races. Next slide.
We hypothesize that although Black men are at greater risk of developing prostate cancer, they remain underrepresented in prostate cancer research. Next slide. To complete our research project, we identified the quantity of black cell lines available on the ATCC and Millipore Sigma website. We located and observed the amount of literary sources that utilize black cell lines and investigated the total amount of Black men voluntarily participating in prostate cancer clinical trials.
Okay, demographics of cell lines available. So these graphs on the screen represent the distribution of available cell lines on both the ATCC website and the Millipore Sigma website. On the ATCC website, there are a total of 22 cell lines available. Of these, 17 were acquired from Caucasian men, 1 from a Black man, and 4 had unspecified races. Whereas on the Millipore Sigma website, there were 7 unspecified races cell lines, 1 from a Black man and 3 from Caucasian men. In total, there were 33 cell lines available, and of that, only 2 represented Black men, which raises the question of why are many Next slide. Okay, so clinical trials findings. So we reviewed 393 clinical trials, and of these, only 198 had racially identified its participants. Of the 198, there were a total of 77,028 participants. Only 8,408 were Black men. That is 11%. We also looked at the percentage of involvement of Black men based on each study, and only 4 studies had 50% or more Black men involved. Again, this emphasizes how little Black men are involved in these clinical trials. Next slide. Okay, so results from the literary paper. So this part of the research was the hardest for my partner and I because we could not have done randomized search, because if we had typed in Black men and prostate cancer or anything of the sort, we would get papers on racial disparities rather than actual prostate cancer research involving Black men. What we had to do was to more specify our search, and we had to create the name from the ATCC website of the Black cell line and input it in the search bar for PubMed, where we were able to find 2 papers. One which spoke of how RNA can prevent prostate cancer cell growth, and the other which spoke of 17-alpha estradiol inhibits prostate cancer cell growth. So again, cell lines, like there's 2 cell lines available, but they're not really being used as much. Next slide.
Our research suggests that there is significant overrepresentation of Caucasian men in prostate cancer research. However, it is evident that men of African descent have higher risk of prostate cancer, which is anomalous to their representation in research. Black men are constantly underrepresented in prostate cancer research, yet they suffer a greater burden from the disease. This gives insight to the apparent racial disparities in prostate cancer research, which lack diversification in tested subjects. As shown in the previous results, there is not enough experimentation conducted with Black test subjects. Next slide.
Impact. So in completing this research, we hope that we will be able to raise awareness about how greatly underrepresented Black men are in research regarding prostate cancer, and also hope to highlight the importance of diversifying our research sample in order to better tackle the evident racial disparity. For future directions, we hope that we will be able to conduct the same study with the same procedure, except we'll be looking at breast cancer instead to see if Black women with breast cancer suffer the same fate as Black men with prostate cancer. Next slide.
Now at this time, before we take any questions, we would like to thank our funders, mentor, and the ECS faculty staff members for making this opportunity possible. Next slide, please. If anyone is interested in knowing more about our research, you can contact us via email at brieprom756@outlook.com and brittanyfulvia_anderson1@gmail.com. Now time for questions.
Okay. So the first question is, so you mentioned that obesity increases a male's risk for developing prostate cancer. Is it possible that African American males are more likely to be overweight and therefore more likely to have prostate cancer?
I guess you could say yes. They're more likely to be overweight if it comes to the fact that like they don't really eat as healthy because expensive food. Healthy foods tend to be more expensive. So considering that if they eat a lot of like fried food and stuff like that, the chance of being obese may increase. So yes.
And dieting was a means of prevention as well.
Okay. What are some possible reasons that Black males are underrepresented in cancer research?
Well, some of the reasons we looked at was the fact that there is some level of mistrust of Black men in the medical system.
So because of this mistrust, the mistrust would be based on like past events that happened in throughout the history, such as the Tuskegee case and Harriet Lacks case. So all these are a reason which contributes to the great level of mistrust that Black men may have within the medical field.
Okay.
So, yeah.
Okay, Dr. Stanford has a question.
Hi, good afternoon.
Good afternoon.
I was just wondering, are African American men or African Americans in general at higher risk for a lot of diseases or most diseases? Or a certain type of disease?
I would say yes, because I had read this paper talking about poor prognosis of African Americans and stuff like that, and it was mainly directed towards the cancer. But considering like some of the reasons behind it, I would say that it would like continue more into other diseases as well, because a majority of studies tend to be mainly Caucasian, or like either primarily or entirely Caucasian. So I'd assume that that would be one of the factors affecting it.
Okay, thank you.
Thank you. Okay, so we'll get ready to move to our last presenter. Adriel. I'll load up the slides right now.
So, oh, thank you. So good afternoon, everyone. My name is Azza Williams. I'm a junior psychology major on the St. Croix campus and a Mark Scholar with the Emerging Caribbean Scientist Program. For the past year, I've been doing research with my mentors, Dr. Alita Bauman from the University of the Virgin Islands, Dr. Karen Schoen, and her graduate student, Michael Rosario from Boston University. And what I've been looking at is the relationship between subjective social status, perceived discrimination, late life depression, on executive functioning in older Afro-Caribbean adults. Next slide, please. So what do any of the words in the big words in the title mean? I'm glad you asked. Let's go through it. So executive functioning is defined as a collection of higher level cognitive processes that take place in the frontal lobe. A lot of these processes you know already, but don't know the name to, such as inhibitory control, which is just self-control, working memory, cognitive flexibility, which is you can switch from thinking about one concept to another concept and switch back and forth. And the most known for the frontal lobe is logical reasoning. So as people get older, the executive functioning starts to decline along with processing speed. So this means that the functioning processes like inhibitory control, logical reasoning start to decline, but also what starts to decline is how quickly people can complete the task using executive functioning. There is little research in older Black people, which will be a trend in research, as you will see. Next slide. Subjective social status is defined as an individual's perception of his own position in a social hierarchy, and social hierarchy is not labeled as just one thing. It can be as small as a family, as big as society itself. SSS is considered to be a part of an individual's overall socioeconomic status, with SSS being associated with other measures of socioeconomic status, such as education, finance, and occupation. What's important about SSS is that it allows researchers to look at how subjectively how people see themselves in their socioeconomic status with finance and education. It's all about subjectivity. And SSS has also been found to be related to race and marriage. So if you're married, you're more likely to have a better subjective social status. And it's also based on race. Next slide, please. Perceived discrimination is the individual's perception of the negative detrimental treatment of different racial, ethnic, national, or other groups. Perceived discrimination also has been found to have an inverse relationship with cognition in older adults. So as someone has a higher perceived discrimination, if someone been discriminated more, they perceive that they've been discriminated more, their cognition actually goes down, or they have less cognitive abilities than someone who has not. Again, little research in older Black people. Next slide. Now, late life depression is defined as a major depressive episode that happens to a person that is at least 40 years old. Now, this definition of 40 years old is not stationary. Late life is a wide range of numbers, but for our intents and purposes, it's at least 40 years old. And late life depression has been found to be related to the denigration of executive functioning in older African Americans. So not just if older African Americans have late life depression, but they've also been found that the severity of the symptoms can speed up how fast executive functioning denigrates. And again, limited research in Black people. So my hypothesis, for the research question, I wanted to ask, does subjective social status moderate the relationship between perceived discrimination and depression on executive functioning in older Afro-Caribbean adults on St. Croix? For my alternate hypothesis, null hypothesis is that the independent variables, which is subjective social status, perceived discrimination, and late life depression, do not predict executive functioning on older Afro-Caribbean adults on St. Croix. And my alternate is that they do predict. Next slide, please. So about the participants. The participants will be 60 Afro-Caribbean men and women aged between 50 to 80 years old. There are a couple of things that need to happen in order for them to be eligible, though. They need to be non-smokers, fluent in English, or fluent enough to complete the instruments. Identify again as African American, Black, or Afro-Caribbean. They shouldn't have a neurological disorder, or shouldn't they should reside in St. Croix for the better part of 20, 30 years. And the vision needs to be able to be corrected with glasses or contacts. And next slide, please. So these are the instruments that I will be using for the study. For subjective social status, I'm going to be using the MacArthur Subjective Social Status Ladder, which is just a ladder with 10 rungs on it. At the bottom, the lowest rung is 1, and that is the worst in society, and the highest rung is the best in society. So we just asked the participants, where do you see yourself in regards to your community and society? So it's a two-pronged question. They ask about society and community. And we just base it off of that. No. I'm not on that yet. Thank you. Late life depression is a geriatric depression scale, and it's a basic depression scale that asks about different types of symptoms. Perceived discrimination is the experience of discrimination, which asks about interpersonal discrimination and non-interpersonal discrimination. And executive functioning is chill making test B, which is just asking about if you have any type of, which is just a test. Yeah, you guys are fine with that. Next slide. So COVID-19 disclaimer, as of March 12, 2020, in-person data has been suspended at the Boston University and United States, the University of Virgin Islands due to COVID-19 pandemic. So we are not able to collect any human research. Next slide. So currently we have 6 participants that completed the cognitive and behavioral health battery of tests for the study. 3 of those participants have completed the status, and only 1 of the participants overall was ineligible to proceed to the subjective social status questionnaire. Next slide. And these are my references. Next slide, all the way to the end. Okay, there. So thank you. I want to say thank you to Alzheimer's Association, Boston University, UBI, the Emerging Caribbean Scientist Program, Dr. Bauman, Dr. Schoen, Dr. Michael Mazzario, Dr. Teresa Turner especially, and my partner, research assistant, Alexandre Carr. So any questions?
Okay, thank you. So I do have 2 questions for you. What do you think is a possible physiological connection between, perceived discrimination and cognitive decline?
Like that's Dr. Bauman. One possible. So there's this thing that I do with my mentors, which is a journal club, and the journal club actually, one of the articles that I presented talked about the fact that white matter in the brain due to water in there may be a reason why we see a decline in executive functioning. And what was the other part of the question? Perceived discrimination, I believe?
Yes, so yeah, what's the connection between that and cognitive decline?
So that may be, we may see like less white matter because some parts of the brain aren't being used when someone is being perceived as being discriminated. Another thing is that the hippocampus, which is what the research that the bigger part of my study is doing for Dr. Schoen, is that they're looking at the hippocampus. And that may also be a point in, you may not use the hippocampus as much, or something may be wrong with your use of the hippocampus if you're being perceived discriminatorily. But I haven't done that much research on it as of yet, because I'm still writing my paper that is going to be published, hopefully. So if the person who asked that question, I can get back to you when I have actually done that research.
Okay, and then how reliable do you think the subjective social status scale responses are?
So the subjective social status responses have been found to be reliable. I think the Cronbach alpha is like 0.8, which is good. I do not know if it's going to be reliable or valid for the St. Croix, because it has not been used on St. Croix as I know it, which is why one of the reasons why I'm so interested about this study is because a lot of the instruments that you heard me talk about has not been found to be valid or reliable down here, and I hope to be the one to do that.
Okay. Thank you.
Thank you so much.
So Deidre, are you there? Could you hear us?
Yeah, I'm here.
Okay. Do you have your slides?
I have, because I presented this week for my Rutgers RU yesterday, and we only had to prepare a poster. So I have like a poster with a video over it, and I could talk over my poster as well, but it's not slides, if that's okay.
Okay, yeah, so we'll let you go now. You could go ahead and share your screen. And
Okay, I'm gonna share my screen now. Okay, can you guys see and hear me this time?
Yes, we can.
Okay, so good afternoon, everyone. My name is Deidre Lee, and this summer I worked in a virtual RU program at Rutgers University, and I was in Dr. Jeffrey Zahn's lab working on a project called Finite Element Modeling of the Electroporation Phenomenon using microfluidic devices. So what is electroporation? So electroporation is a commonly used non-viral method for intracellular drug delivery of biomolecules and therapeutic agents. And this is a big research topic in immunoengineering and drug delivery in general. So electroporation works by applying an electric pulse that induces this difference in electrical potential, and that causes in temporal pores inside the cell membrane. So it makes it more permeable for the access of molecular delivery into the cell. So with this technique, it's crucial to obtain efficient delivery without compromising the cell. So basically in figure 1, it shows that the phosphobiolipid layer and the voltage is applied, and this potential difference causes the biolayer to cleave and form like these pores, and it's supposed to be reversible. So that's the technique. You have to be able to apply just enough voltage to create these pores, but not enough that you make them permanent and the cell dies. So a microfluidic platform allows us to electroporate many cells at once in an automated and controlled environment while maximizing cell viability. And computational models are crucial in the design process as they identify microfluidic design for optimal results. And that was basically my project, was to design a computational model of cell interactions with an electric field and the cell membrane becoming permeabilized when we change the membrane resistance. So I did my computational work in COMSOL Multiphysics software version 5.4, and I started off doing tutorials known as the electric field from a charged sphere, a controlled micromixer, and a contact impedance. And these tutorials just helped me get acquainted with the software because it was new to me, and also the skills gained from these tutorials compiled together would basically help me solve my problem. So figure 2 shows a mesh plot of the charge sphere that I got out of the first tutorial, the electric field from a charged sphere tutorials. And the second, figure 3 shows the voltage field that surrounds this electric field supposedly, and is done by contact impedance. And figure 4, I don't know why it's labeled figure 3 here, but yeah, shows the velocity field. So this demonstration should kind of be like how the microfluidic device looks like, a flow of a substance through, and it has an electric field through it and should electroporate the cell with this flow. And so because we were only like a 5-week program, I didn't get to the extent of compiling all of these results together in COMSOL, but that's like technically my future work and what I'll be continue working on, hopefully for the remaining of the summer, is just to generate a cell in figure 2, apply a voltage around it and verifying it using the equation voltage over length. And we use the properties in COMSOL to just verify the transient voltage, because doing these computational works, we have to do a lot of verification processes because we want to make sure that they actually work, and that when we take it to the experimental table, that it works how we think it would. And then we would verify certain angles and the conductivity for the electroporation of the cell. Again, I didn't get a lot into these parts. I can't really explain too much, but yeah, so the end result, what I'm hoping to get is to demonstrate a cell being electroporated in this flow, in this constant flow property. And then later on, we'll verify it with the help of my grad student and hopefully apply it to like microfluidic properties. So really design the microfluidic device. And I would like to thank my PI, Dr. Defrizan, his master's student, Christopher Kidjob, and one of his undergraduate students working in his lab, Upasana Ghosh. We had meetings every Friday to work on our project, and the NSF, the RU. RISE was a big program, and I was in the cellular bioengineering RU section. And I would like to thank RISE at Rutgers as well, and for you guys for listening. So thank you so much. Do you have any questions?
Thank you. So we do have a few questions. So how do you know what the best voltage for electroporation is?
It depends on the diameter of the cell, and using these equations. I have one here, but there's a lot more extensive equations in my list of properties. So it depends on the shell. So the end goal is for me to have blood cells. So I guess the dynamics of the blood cell, sorry, don't know why. Okay, just so you can see it. Yeah, so I'll be working with blood cells, so I'd have to go into detail about the cell diameter of the blood cell, and then calculate the best voltage to apply to the cell without killing it. Yeah.
Okay. Yes, and then I guess the follow-up to that would be, do different cells have different preferred voltages?
Yeah. Yeah, because different cells are different sizes and have thicker membranes. So we have to take all that into account.
Consideration. Okay. Do you think it would be useful to electroporate tissue as well for bulk delivery of drugs instead of using individual cells?
Bulk delivery, I think that's the end goal. The whole idea of having the microfluidic platform is that we can electroporate multiple, I know it's just multiple cells at once. I'm guessing a combination of cells is tissue, right? So I'm assuming, yeah, we can do that. And I'm sure Dr. Zan has papers on that as well. But I was just working on an individual cell, just the basics, because you won't have to start at the basic of electroporating one cell and then making it bigger. But he has shown that in this microfluidic platform that he has electroporated a lot of cells at once. Yes, to maximize drug delivery. Yeah.
Okay, thank you. So I'll open up the floor. Are there any other questions? You can go ahead and unmute and ask any questions if there are any more. Okay. If not, we can go ahead and wrap up. So I just want to say awesome job to all of you guys that presented. Great work, and it was definitely displayed here. I also want to commend you guys for adapting to a virtual experience and still making the best of it. So we want to acknowledge the National Science Foundation HBCU Up Grant. We want to acknowledge NIH RISE and MARC grants. Also the College of Science and Mathematics, the Emerging Caribbean Sciences Program, and the generous donations of our private donors. We also want to say thank you to all the mentors for your commitment and support to the students and for contributing to undergraduate success. Thanks again for being here and showing your support to all of the students. And we've made it to the end of our first virtual summer symposium. So thank you all again, and I hope everyone enjoys their weekend.
Thank you.
Thank you.
Thank you.
Welcome. All right, goodbye.